Mitosis reading. Mitosis is the division of the cell nucleus. During mitosis, the chromosomes in the parent cells divide into two identical sets. This occurs prior to the division of the cell itself. In cells, mitosis is the process by which two identical cells are created from one. Organisms need to reproduce new cells to grow and to repair damage to existing cells. The cell cycle. The process of mitosis is part of the cell cycle. The cell cycle is the sequence of events that a cell goes through over its lifetime. The cell cycle is divided up into three events. The three events of the cell cycle are interphase, mitosis, and cytokinesis. Interphase is a period of cell growth and development. During this phase, the cell is carrying out its normal activities. If the cell is a bone cell, then the cell is making bone. If the cell is a stomach cell, then it is secreting digestive enzymes. At the end of interphase, the cell is ready to divide into two. Mitosis is the division of the nucleus and the genetic material it contains. Cytokinesis is the division of the cytoplasm and its organelles into two daughter cells. Interphase. Cells spend most of their lifetime in interphase. However, the actual lifespans of eukaryotic cells vary. In ideal conditions, a yeast cell lives for about 2 hours before it divides and forms two new cells. An amoeba in a laboratory culture may live for a few days before it divides. In animals, some cells of the early embryo may divide every 15 to 20 minutes. In multicellular organisms, many cells remain in interphase permanently. Biologists have identified three distinct phases in interphase. The three phases of interphase are called G1, S, and G2. During G1, the cell is carrying out its normal activities. In the S phase, the uncoiled DNA in the nucleus known as chromatin replicates. In the G2 phase, the cell membrane and the cytoskeleton undergoes rapid growth that prepares it for mitosis. Cells that stop growing and dividing remain in the G1 phase. The nerve cells in the brain and spine of animals are incapable of dividing and so remain in the G1 phase throughout their existence. Mitosis in these cells would interfere with memory. In dividing cells, interphase immediately precedes mitosis. Phases of mitosis. For cells that divide, the second event in the cell cycle is mitosis. Mitosis is a continuous process that scientists divide into four phases: prophase, metaphase, anaphase, and telophase. As you read, keep in mind that mitosis produces two identical nuclei with the same number of chromosomes as the parent cell. Prophase is the first phase of mitosis. During prophase, as shown in the diagram, the chromatin, uncoiled DNA, which has already replicated, coils and forms chromosomes. The genetic material in the cell becomes visible at this time. Simultaneously, the nuclear membrane breaks down and disappears. Metaphase is the second phase of mitosis, where the chromosomes are moved to the center of the cell. Anaphase is the third phase of mitosis. First, the centromere of each pair of chromatids divides. Then the chromatids separate and move toward opposite poles of the cell. The movement of chromosomes is rapid and dramatic. Telophase is the final phase of mitosis. At the beginning of telophase, the two identical sets of chromatids are clustered at opposite sides of the cell. During telophase, the chromatids unwind and elongate back into thread-like DNA. A nuclear membrane then forms again around each mass of DNA. Cytokinesis. The process of cytokinesis takes place immediately after mitosis. Cytokinesis is the division of the cytoplasm and organelles into two separate cells. The cell now contains the two nuclei formed during mitosis. In dividing the cytoplasm, cytokinesis also separates the other structures distributed throughout the cytoplasm, such as ribosomes, Golgi bodies, and mitochondria. The two new cells that have formed during cytokinesis are generally equal in size. The illustration demonstrates the way in which cytokinesis occurs in cells. References for this reading: Curtis and Barnes, Invitation to Biology, published by Worth Publishers, Incorporated in 1981. Johnson and Raven, Biology, Principles and Exploration, published by Holt, Reinhardt, and Winston in 1996. Tolley, Modern Biology, published by Holt, Reinhardt, and Winston in 1989.